Hi, it's Aiden Corvin here from Trinity College, Dublin, and we're hosting the PGC Worldwide Lab meeting today, 18th of October. And our topic is the Pedigree Sequencing Project that we've been doing as part of PGC3. So I will be introducing the topic or giving a little bit of background into the project. And then my colleagues here, Niamh and Collaborators, will be providing just a bit more detail really on where we've got to so far. So we would plan on speaking for maybe about 45 minutes, leaving plenty of time for questions at the end. So if people do have questions, maybe they could keep them till the end. So I'm just going to give it maybe a minute or two just to let people join the call. And if anyone there has done it before, there's an eerie silence that I'm experiencing that I'm not used to on these calls. And maybe that's just the sacrifice you make when you're presenting. But anyway, so I can't hear anybody out there, but I will start now in about a minute's time. So, obviously, as we all know, significant progress has been made in the last decade in identifying risk loci for psychiatric disorders. And the PGC has had a fundamental role in this. And I think this has been for two reasons. The first reason being that it's facilitated analysis of samples with sufficient power to identify common variants of small effect. And also the framework that it's provided has allowed a methodologically rigorous approach to provide robust results. And it's not only providing the results of the pipelines and the analysis, but it's also making these results available publicly so for secondary analysis and other projects. So that's been a real strength of the PGC. As I've mentioned already, most of the identified loci have small effects. So I just highlighted the schizophrenia paper there as a poster boy, really, because in schizophrenia, we probably had access to larger sample numbers earlier than the other disorders. But actually, as time has gone on and the numbers have built from other conditions, there's a lot of loci being identified as well. I mean, one of the issues that we face as a field is that the effect sizes we're finding are small. So it's very difficult to extrapolate biological meaning or understanding from small genetic effects. And even though CRISPR and other techniques have been developed, making it easier to create model systems, it's really only possible to model a few loci at a time. So essentially, what you're trying to do by modeling small effects is it's a bit like trying to understand a concerto and the full impact of an orchestra from just sampling a couple of violinists. So really what you need, you need a more complete view of what's going on, or maybe a different view. So one of the things that we know across disorders now is that for most conditions there's a complex genetic architecture. So most of the discovery that has been done to date has been done for common variants, though I think for more severe neurodevelopmental disorders like autism, schizophrenia, probably bipolar as well, there's been more work, there's increasing amount of work done on the rare variant side, and it's clear that there's a spectrum of variants from common to rare that contribute to risk. And I think the other thing that is really quite unexpected as a clinician who started out in this field is the degree of overlap between conditions that we're seeing. So this is a slide that I think is well known to people, diagram from the Brainstorm Consortium paper, really just showing the extent of overlap between the different conditions that we look at. And this is interesting, but also, as I said, wasn't appreciated at the time. So in the original molecular genetic studies, as people started to put together family collections and do linkage studies, the focus was very much on recruiting quote-unquote pure families, where you were just seeing segregation of a single disorder. And one of the things we've wondered about in contemplating this project is if there are families out there where there's kind of more mixed phenotypes, and particularly phenotypes that have a neurodevelopmental flavor, where there might be schizophrenia or ASDs in a family and that they may be valuable for identifying rare variants. So we know from the literature so far that zone A, the zone of large common effects, is empty really for neuropsychiatric disorders. That doesn't really apply. Most of the progress we've made has been in zone B. And the interest in this project really is about moving into zone C and looking for rare variants which may have larger genetic effects. And they may be useful, and we know this from other common conditions, that finding rare highly penetrant mutations can be quite useful in understanding the biology of a condition and developing new drugs. So really it's this zone C that we're looking at, but it's challenging. So here is a slide from a very nice paper by Zuck and colleagues. It was in PNAS a couple of years ago. And what you're looking at here is the colored lines are essentially odds ratios. So you move from an odds ratio of a genetic effect from 1.5 up to something that's 20 on the right-hand side in blue. And what this really is a power calculation. And the power calculation is at the level of the genes. So what Zook and colleagues did here was they collated any mutations that were in a given gene. So the gene was the unit of analysis, and they were trying to calculate the power of different study models. So the S at the bottom here is selection. And so if you look at the 10 to the minus 3, that would be weak selection, where common and rare variants are of roughly similar, or can be of roughly, get up to sort of similar frequencies over time in the population. This is weak selection. And as you see, you know, you're in the range of cases of 10 to the 4, 10 to the 5 for effect sizes, you know, that are relatively small. The challenge arises where you move to the right, down to 10 to the minus 1, when you're looking at strong selective effects. And in this model, the numbers just become staggering, and it becomes very difficult to consider powering rare variant studies. So this is a real challenge. So there's an interest in identifying rare variants, but actually the association methods may be quite punitive in terms of the sample numbers required. So what we have been interested in, and this is an image from a paper by Jim Lupsky a couple of years ago, and the image is about a concept that they describe as clan genomics. So essentially, what you're looking at is the human population as a family tree. And what you're seeing is that as you look at new mutations, de novo mutations in sort of the red moving into the orange, the mutations that have large effects on risk are selected against over time in populations. So they're going to be present for maybe a few generations within a family, but over time then they'll be selected against. So you go through the colors of a bruise, the yellows, the greens, the blues, as you get down to sort of more common effects that are, you know, don't have the same selection pressures. So the idea was that there's a lot of value in looking at family structure at potentially being helpful in identifying what were de novo mutations in many cases, or mutations that are only surviving for a small number of generations. And there is practical support quite recently from the schizophrenia genetics literature for this view. So you see that the DURVs, which is disruptive ultra-rare variants, from the Genovese paper, which is one of the largest schizophrenia exome studies published to date, there was an excess of these disruptive ultra-rare variants in schizophrenia cases compared to controls. There was also evidence in that paper that most of these were likely to be inherited rather than de novo, which would be in keeping with the model we've been interested in. And I think helpfully, and this is something we do need to consider, is that most of the genes affected were brain-expressed synapse-related genes. So I think using different methods, as we get a better sense of molecular pathways that might be plausibly involved in these conditions, it reduces the search space from all genes down to a subset of genes. So that the aim of our study is really to identify and analyze large, densely affected pedigrees with psychiatric disorders. We were interested in not just pedigrees that are, quote, pure pedigrees for a specific disorder, but just where there's a heavy concentration of psychiatric illness. We wanted to contribute to a consensus on methodology and pipelines for the analysis of whole genome data in pedigrees using, you know, polygene risk score, SNV and CNV methods. And we felt this was important because reviewing the literature as we started this project, there's a lot of different approaches being applied, and we felt it would be important to have a consensus on and develop a pipeline which could then be applied by others. And, you know, we obviously were keen to make our data available to the field. So part of the project is to look at the individual pedigrees we'll talk about now, but part of it is to collate the data and provide a resource to the field to contribute to gene discovery. So I'm almost at my last slide. So this is really just a list of the collaborators and just describing the main diagnosis in each of the pedigrees they've provided. And you can see from the slide that some of these pedigrees have quite a range of diagnoses. So for example, the large pedigree from Muhammad in Pakistan has schizophrenia, depression, anxiety disorders, and epilepsy present. So we've been lucky, we've had great collaboration so far, and a number of our collaborators had already got some whole genome or whole exome sequencing done, and we went back to the pedigrees and augmented that with some of our own sequencing. So thus far in total, we've looked at, we have sequence data, I should say, on 49 pedigrees and 472 individuals. And we're working through those numbers. And as I will say again at the end, if anyone is interested in working with us and has pedigree data, we'd be delighted to talk to you to work on this. So that's really an overview of the project, how it came about, and where, you know, the samples come from, etc. So I'm going to hand over to Niamh now, who's going to talk to you about some of the single nucleotide variant work we're doing, and then Cathal is going to talk about the CNV work. So over to Niamh.
Nice. Yeah. So hi, I'm Niamh, and I'm the postdoc on this project. And today I'm just going to be focusing on one sub-branch of our bigger pedigree analysis. But before I go into details on that, I just want to make a comment about the different types of pedigrees that we have. So whilst one of our aims is to develop standardized approaches and pipelines for analyzing whole genome sequencing data from pedigrees, the pedigrees that we're working with are definitely not standardized pedigrees, and there's a lot of variation there. So for example, we have on one hand small pedigrees like this, where we have one single generation with a large number of affected siblings, going to what is more traditionally seen in linkage analysis studies, a 3-generational pedigree, to some examples of larger pedigrees with a big mix of disorders within the pedigree, with some examples of consanguinity, going to even larger pedigrees with a variety of phenotypes, and even more inbreeding loops. And what this really shows is that there's no one-size-fits-all for analyzing these data sets. So whilst we're making standardized approaches, we're going to be having tailored analytical pipelines for each of these pedigrees. So the pedigree I'm going to be talking to you today about is a Tourette syndrome pedigree from Costa Rica that we are working on in collaboration with Carol Matthews. This is the pedigree down at the bottom, and I'm sorry that it's so small, but this is the only way I could get the entire pedigree onto one slide. This pedigree contains over 500 individuals and spans over 6 generations. At the top of the pedigree, we have 6 different founder pairs. And at the bottom of the pedigree, we have our affected individuals who are highly divergent, most of whom are separated from each other by a minimum of 10 meioses, apart from one parent-offspring pair that we have. So these individuals, we had 20 individuals, 19 of whom passed whole genome sequencing. So we have data on 19 individuals, 16 of whom are affected by TS, 9 of whom are co-affected with ADHD. 3 are unaffected by TS, but one of them is affected by ADHD and another is affected with OCD. So because these individuals are so unrelated and cover such a great genetic distance, the study design I chose to use to analyze this pedigree is identity by descent analysis. And just to explain a little bit about what identity by descent analysis is, if you look here on the left, we have a graph looking at the number of meiotic steps separating individuals against the median length of shared segments. And, oh, I managed to go backwards, so let's see if I can go forwards again. Yeah, so on the left-hand side of this graph, where you're looking at less people who are unrelated, so population data sets, association studies have been very successful at identifying common variants that are associated with phenotypes of interest. When you reduce the number of meiotic steps separating people, there are additional methods at our disposal for analysing and looking for variants associated with illness. On one of these being looking for regions of identity by descent. So if you look on the figure on the right, there's an example of two half-siblings who share a common mother, and these siblings share 50% of their DNA identical by descent. And you can see that some of it is coming from one chromosome copy in the mother, and the rest is coming from the other copy. And so we don't, rather than getting the whole copy transmitted from to each offspring, recombination has occurred. So there's different lengths of IBD sharing across these individuals. When you look at people who are more distantly related, like the individuals on the right-hand side who are fifth cousins, these individuals are separated by 10 meiotic events, 10 meiotic steps, which means that the chances of having regions shared between the two that are identical by descent is much, much smaller. And in fact, fifth cousins usually have no detectable IBD sharing, but when they do, it tends to be a single region that's around 8 megabases large. When you increase that further and go up to 25 generations separating individuals, you expect to have no IBD sharing in general. But if you do see a block of IBD sharing between individuals, it tends to be much shorter and about 2 megabases long. And then if you want to look, that's just between two individuals who are related to each other. If you then try and compare to 3 fifth cousins who are equally unrelated to each other, the chances of finding the same region shared across 3, the chances of that are much, much smaller. And that is the method that we are using to try and identify regions that are shared between our affected individuals, with the idea being that they are so unrelated that if they do have regions that are shared between each other, that this region is going to contain variants that are important for their phenotype. So we are working with whole genome sequencing data, which hasn't really been used for identity by descent analysis. Most commonly, people have been using GOS chips. So our data set, our unfiltered pedigree VCF file, contains over 13 million variants, which is far too many for a identity by descent analysis. There's also the fact that if you have rare variants in there, they're not going to come up on a common ancestral haplotype. So what I first did before performing this analysis was to filter the data on standard QC filters in Plink, removing things that were very rare and LD pruning down to 0.6, excluding Mendelian errors and looking only at SNPs, SNVs that have RS numbers. This reduced the size, the search space of our data set from 13 million to just over 500,000 variants. This next slide shows a graphical representation of the pipeline that I put together to analyze these data. The first step I did was I performed haplotype phasing. I did this using ShapeIt2 and a hidden Markov model extension, which was published by O'Connell et al. In 2014. This particular method is useful for our analysis because it allows you to incorporate both a population reference panel and pedigree information to improve the power of haplotype phasing and reduce switch error rates. After phasing the haplotypes in the pedigree, I then performed pairwise IBD to find regions of the genome that were shared across each pair of individuals. And the thresholds that I used for this was looking for IBD segments that were greater than 1 megabase and that had log scores greater than 3. When I generated this list of pairwise IBD segments, I then performed a multi-individual IBD clustering analysis using the efficient multiple IBD tool. And this resulted in the identification of over 450 multi-haplotype IBD clusters. So these were regions of IBD sharing in 3 or more individuals. Of these, 30 of the IBD clusters were shared across 4 or more haplotypes. So I then, which was an encouraging amount of data to be looking at. So I then took the multi-haplotype IBD clusters and subset them by founder pairs. So what do I mean by founder pairs? If we just go back to the original pedigree, you'll remember that I said that at the top of this tree, each of these individuals at the bottom is related to different combinations of the people at the very top. So this figure on the left just shows of my 19 individuals who is related to which founder pair. And as I have individuals who are not related to each other, do not share a founder pair ancestor, I decided to, rather than look across the 19 individuals, look for regions of IBD sharing that were clustering with one specific founder pair or another. And once I identified these enriched clusters of IBD sharing, I then took the regions that were interesting and mapped the IBD segments back to the base chromosomes. And this was a really important step because it allowed me to do a couple of different things. First, it allowed me to confirm the IBD sharing in these individuals to make sure that there were no spurious findings coming through. Secondly, it allowed me to refine the boundaries of IBD sharing. One of the caveats of using refined IBD for pairwise IBD analysis is that it doesn't allow for any genotype error. So if you have a variance that is different in one individual but is within a common ancestral haplotype block, refined IBD might consider it to be a recombination breakpoint and throw that person out or exclude it from the analysis, or shorten the boundaries of the haplotype region that is shared by individuals. And thirdly, this links in with the inability to cope with missing genotype or genotype errors. I wanted to go in and see if there were any additional individuals who were sharing IBD in these regions who might have been thrown out because they had one or two variants that were different, which is very plausible when you're dealing with whole genome sequencing data compared to genotype data. In the interest of time, I'm just going to focus on two examples of regions that I identified that were identical by descent in this pedigree. The first is a 5 megabase multi-haplotype cluster identified in 3 individuals who are descended from founder pair D. So this is 3 out of the 5 individuals who are descended from founder pair D. And the second is a 1.2 megabase multi-haplotype cluster identified in 4 individuals descended from founder pair C. So this is half of the individuals descended from founder pair C. This first example, the 5 megabase cluster descended from founder pair D, is on chromosome 4. And what I've shown before at the bottom here is on the red bar represents the EMI predicted multi-haplotype cluster, roughly 5 megabases, present in individuals 1, 2, and 6. But when I took when I went back to the phased haplotype files from SHAPEIT and manually analyzed the data further, I could see that actually, whilst EMI was quite strict with defining the boundaries of this haplotype region, that in actual fact, the region shared identical by descent across these 3 individuals is much, much bigger and actually spans more than 11 megabases. So this is quite a large region of IBD sharing for people who are so unrelated, and therefore is a very interesting region to follow up further. The second region I want to talk about is the 1.2 megabase region on chromosome 3. So this is represented by this orange block up on the top. And was predicted by EMI to be shared across 4 individuals, individual 1, 5, 6, and 18. When I took the coordinates of this region back into the phased SHAPEIT output data, I found a couple of interesting pieces of information. Firstly, that individuals 5 and 18 share a region that's much, much bigger than the limited 1.2 region identified by EMI. And on top of that, there was another person, ID 15, who's also descended from founder pair C, who is completely identical by descent. She has the exact same haplotype with 5 and 18, and that these boundaries are much, much further away than the boundaries predicted by EMI. In addition, the boundaries, the left-hand boundary was very correctly identified by EMI for individuals 6 and 1. So that seems to be that break point seems to be driving the left edge of the IBD haplotype in EMI. But actually on the right-hand side, there were much longer regions of haplotype sharing in these individuals. Lastly, I've highlighted in purple here, when I was looking at this phase data, I identified 2 additional individuals who do not share this region found by EMI, but in fact share a small region on the right-hand edge of this haplotype. So in actual fact, there is a region, a small region, which is why it didn't show up in EMI, but an 800, just over 800 kilobase region shared across not only the original 4 people identified by EMI, but an additional person descent from individual C, founder pair C, and 2 individuals descended from founder pair D and founder pair B. This is interesting because even though they are, even though we think that they are, we have them down as being unrelated people at the very top of the tree, this family comes from an isolated region in central Costa Rica, and chances are that actually the founder pairs at the top of the tree are probably related to each other at some point even further back in time, but we don't have the data to show this. So it's not surprising that we're seeing potentially IBD sharing across people from other founder pairs as well. So this region looks really interesting to have an 800 kilobase region shared identical by descent across such a large number of people, and we will be following this up in more detail. So to conclude, we have identified multiple regions that share identity by descent between distantly related individuals, and we will be investigating the genes and variations within these regions further to identify those which may be contributing to Tourette syndrome risk in the family. Our future directions, I mentioned that pairwise IBD doesn't allow for genotyping error, so or differences in genotype. So one of my plans is to do pairwise IBD share to look at relaxing the pairwise IBD sharing thresholds to see if I can identify shorter regions of IBD sharing that are potentially present in even more individuals. Once I've done that, a crucial part of this is that what we could be identifying, whilst it's shared across the cases, it could also potentially be common in the population that they're coming from. So it's very important to go back and look at a population relevant control cohort to test for the frequency of these haplotype regions in the general population and perform a case control association. After that, I'd also like to perform CMV analysis on this pedigree using our in-house pipeline that we've developed that Cathal is going to talk about next. So I'm going to hand the mouse over to Cathal. And if you have any questions on what I've said, we can discuss it at the end. Thank you. Oh yeah, that's actually probably easier.
So thank you very much, Niamh. So hi, my name is Cathal Lormond. I'm a PhD student in the group, and I'm going to chat to you today about work that myself and Niamh have been doing jointly on putting together our in-house pipeline for calling copy number variants from our family-based whole genome sequence data. So, as I think a lot of us know already, copy number variants, or large deletions or duplications of the genome, are quite well known to have a contribution to the genetic etiology of psychiatric illness. Certainly for schizophrenia, we know that rare CNVs tend to have a higher effect size than the common SNPs that have been known to be associated. But even so, these CNVs don't necessarily have complete penetrance. So while quite interesting, they certainly provide no end of challenges. So our data is whole genome sequence data, which is fantastic. It allows us to examine a broad spectrum of types of variants from different frequencies across the genome. But to date, there is no best practices pipeline for calling CNVs from short read whole genome sequence data like you might have for SNVs, which is kind of challenging in its own right. But what I kind of want to talk is about some of the pitfalls and some of the challenges we've experienced while putting together or refining these pipelines. So for detecting copy number variants from whole genome sequence data, there's kind of 2 or 3 main methods that people tend to apply. If you have a look at the graphic on the right-hand side, at the top we have read depth-based callers. So read depth-based callers tend to examine if there is a sharp increase or decrease in read depth coverage across at regions of the genome, which might indicate a deletion or duplication event. And for duplications, the proportion of increase in the read depth might indicate the copy number status of that region. Another method for trying to detect copy number variants is to look at the actual sequence reads. So typically, you have sequence reads in pairs. So if one pair is mapped to a position quite far away from where you expect, then maybe that indicates that there's a deletion or a tandem duplication event. Or similarly, if your reads are mapped in a way that looks like they should be split in half, then that could be an indication of a deletion event. So the two main methods that we looked at were read depth-based callers and combinations of paired density read caller, although it's worthwhile emphasizing that many callers tend to adapt multiple different methods to try and best estimate where we could detect our CNVs. But to date, there is still no one-size-fits-all tool that can detect different types of CNVs from different types of data. So a common strategy that has been employed for a long time is to apply multiple different tools or algorithms and then form a consensus call to accept variants which are found by at least two tools, which is good at giving maybe more higher confidence sites, but really when you apply this, you kind of lose the individual utility or any of the advantages that one tool might have over another, given that there's such a great disparity of detection methods across tools. So our pipeline that we've put together is based really off three main papers. The paper at the top here by Khan et al. Kind of forms the backbone of what we've done. So they applied a consensus method to 91 multiplex families affected with schizophrenia. But one of the main kind of changes they made to a traditional consensus call was to examine these variants which were found by one tool only out of their 4 tools that they'd selected. So the upshot is that, you know, if you have a variant that was found by one tool, but also that variant was found in a close family member, well, then maybe that's sufficient evidence for you to retain that variant as a high confidence, where we would have thrown it out beforehand. So the other 2 papers, we kind of informed on different tools we might use or different tips and tricks or stuff to be aware of while you apply, while you look at whole genome sequence data or while you look at detection from this data. So we've amalgamated a unique combination of tools, and we've taken some ideas from the others and then kind of to maybe better empower our pipeline. So an idea we took from the TROS paper was to examine just the individual calls, the calls from each of the individual tools first, maybe to see what they might be telling us. And what we observed was that, for example, if you imagine these light blue bars at the top to be multiple deletion calls at a region in the genome for one individual, we kind of thought that this maybe looks like that there are multiple regions, multiple calls getting outputted from each of the tools that actually represent one region. So our first stage of post-processing is that we collapse down multiple calls which actually we think represent the same region. So we've come up with a set of thresholds that kind of look like they might balance out this overrepresentation of certain regions. I should point out that this is much more of an issue with the paired down split read style callers compared to the read depth based callers. And then when we compare across multiple samples, again, the breakpoints for whole genome sequence data aren't necessarily as accurate as they could be. So we might often notice that there would be the same region which doesn't, across 2 individuals, which doesn't have the same start and end position. So what our take on is, which I think is quite a common accepted threshold, is if 2 regions overlap reciprocally by 50%, then we consider them the same region and we consider the union of both calls. So for each individual, our workflow runs as follows. We've selected 2 paired down split read callers, which are on the left in red, Manta and Lumpy, and 2 read depth based callers on the right in green, which are CMB, Nature and ERS. So for each sample, we apply our 4 methods. We collapse down any sites, which again, we think are representing the same region. And we expect that that 2 callers within methods, so Manta and Lumpy and CMB and ERS, should be more similar to each other than they will be across methods. So the first step, we merge these 2 call sets within methods, and the next we merge call sets across methods, because while each have their individual advantages, for some CNVs they should be called by any method, so we can compare across methods. And then finally, we remove any variants which are known to be in repeat or low complexity regions, like segmental duplications, or in pseudo-autosomal regions, or regions of low mappability, like the short arms of chromosome 13, 14, 15, etc. So then, for each family, we can combine all our call sets. Again, even we expect that the regions of interest, or we expect all CNV regions compared to closely related family members, should have approximately the same start and end positions, but just in case we can collapse them down just to represent the same site. Then we can filter out any singleton variants, which is what we call variants found by one tool only, if they're present in only one family member. So after this stage, any variance that is left should have been called by multiple tools, or it has been called by one tool and also found in other family members. Our pedigrees, as you've seen, are quite widely diverse, so we can remove Mendelian errors, and then given a list of databases, we can remove common variants. So our primary analysis then is looking at any variants which might co-segregate with disease, or if any variants are present in known phenotype databases or affecting known phenotype genes. So we're quite happy with our pipeline, but of course we always want to benchmark it. So we downloaded genomic DNA from the very well-known and well-characterized NA12878 individual, and both for parents. We also independently put together a gold standard list of deletion calls for this genome, which was curated from 5 separate resources. Due to the individual performance of each tool for whole genome sequence data, we only examined CNVs that were longer than 1 kilobase pair, which left us with 638 variants in our gold standard set. So in the table here on the right-hand side, we have our method. We applied also the Khan et al. Method exactly as they had it, and then compared the performance of each of our callers individually on this particular gold standard set. So we're quite happy to see that our method performed quite well in terms of sensitivity, which is the proportion of the gold standard sites that were detected. So we detected 86.5% of them, which is quite good to see. It was quite interesting to note as well that the 2 paired-ended split read methods, Lumpy and Mata in red, performed comparably to each other, and similarly that the redef-based callers performed less well, but also comparable to each other. So it's good to see that certainly on this particular data set of gold standard calls that we perform better than the each tool would independently. So we seem to be drawing other calls that might have been missed by each of the individual tools. So to test this out on some of our sequence data, we applied this pipeline to our whole genome data for 52 samples across 9 multi-generational multiplex families, for an average of 5.7 individuals per family. So these individuals were provided by our collaborator, Bill Barley, and they all have CU European ancestry. The breakdown of diagnoses is about 35 schizophrenia, 4 bipolar disorder, 1 major depression, and 12 unaffected individuals. So quite heavily quite heavily focusing on schizophrenia. So to generate the raw sequence data, we aligned and processed it according to the GATK's best practices and aligned to the GRCH38 genome build, and lifting back using the UCSC liftover tools HG19 for comparing some of our common variant data sets. So as a first pass screen, we looked for a collection of well-known CNVs that I have listed here on the table on the left. So deletions, duplications in very well-known regions like the 22q11 deletion, the neurexin deletion. So what we noted was that 1 or 2 of these variants were occurring across the 9 families, which is good to see. They're quite rare, so we wouldn't expect many of them to be occurring in very many families, but none of them were present across multiple affected individuals. None were co-segregating with disease, even allowing for reduced penetrance. So this was nice, just as our acid test to see could we detect them. But one thing that this kind of pointed us towards was originally some of the pitfalls that we can kind of observe with some of the sequence tools. So here is an example of one of the pedigrees that we observed. The individuals marked in black are affected with schizophrenia. Anyone with a green dot is an individual we performed whole genome sequence on. And you can note in the second row under the numbers, there were some individuals who had a deletion present. So we observed that in 4 out of 5 of the affected, there was a deletion on the 3q29, which is very well known to be associated with schizophrenia. It's rare in population, so we thought this is quite interesting. So it's almost perfectly co-segregates with disease, which kind of agrees with what we might know about these large CNVs. It's present in none of the unaffected individuals, so we thought, great, this is really interesting. We dug into a bit and we noticed that this was called by one of our tools, Lumpy only, not called by 3 of the other tools. We thought that's kind of interesting that, this is something that had we been agnostic to some of the family information we might have thrown out, but we retained otherwise because of this pipeline. But it's just, it's slightly a little fishy because this is a 1.6 megabase pair deletion. So we thought it was unusual that some of the other tools mightn't have picked it up. So when we drill down into the raw sequencing data, we noticed that this was actually a false positive. So here is an example of the raw sequence data for one of the supposed carriers. So the red line here is the start point of the alleged CNV, and there's a 10 kilobase, there's 20 kilobase spare window around this region. So these little colored gray, green and blue blocks here represent the raw reads from the sequence data. So what we would expect from this IGV view here is that if there were a deletion to the right of the red line, that we expect almost no sequence reads to be present in this position, which you can actually see there's genomic data on either side of this deletion. So that was a nice quick acid test just to make sure that this wasn't, just to check whether this is a true positive or not. So that was quite disappointing because, you know, on the face it looked great, but it actually led us to have a bit more of a think about how we process the individual tools, because this was found by Lumpy, and what Lumpy can report on is the number of sequence reads which support an alternate event at this site. And the Canadol paper recommended filtering out any calls which have fewer than, for example, 4 calls from Lumpy. But in regions of higher depth of coverage, you can see there's actually, I think, about 100x coverage at this site. If there might have been 4 reads, well, 4 reads out of 100 actually is not quite a lot of evidence. In our case, I think we had 5 reads, so we would have retained this. So this is an example of somewhere where it passed any kind of hard threshold filters, but really we kind of shouldn't be accepting this as a true positive site. So something that we kind of think is a bit more appropriate in looking at regions of maybe higher coverage is to maybe remove variant calls whose proportion of reads which support an alternate event is less than a certain proportion, 25% or something like that. So that kind of it was a bit, you know, it was a terrible Friday to a great Friday and a terrible Monday, but it kind of did really inform us on really some of the pitfalls that you can kind of encounter when trying to detect copy number variants from sequence data. So sequence data can give us a huge range and breadth of information, but it also can bring with it its own errors and misinformation. So we applied our method to our 9 separate families, and here's just a quick table of some of the deletions and duplications that we observed. So we have various different family structures. We have different numbers of affecteds and unaffecteds around each family, and we were focusing here on variants which were greater than 1 kilobase pair and less than 21 megabase pair in length, because some of the paired and split read tools can also detect, well, we'll call variants, putative variants, which actually represent maybe a deletion in the whole genome because they're picking up reads that don't map correctly to either the centromeres or the telomeres. So that's kind of something to be aware of. So we looked at our variants and their overlap with known gene regions, and also for any of our rare variants which overlapped association peaks from the schizophrenia PGC paper. So we noted that we do have rare CNVs which are overlapping known gene regions, and we have rare CNVs which are overlapping association peaks. So this is quite interesting. This is only looking at the variants that are not common based on our databases. Our next step, I guess, will be to look across all the families, given that they have a similar ancestry, and try to find maybe family unique or family private variants, which might kind of give us a better idea of what's happening within the families. You can kind of even note for some of the families that there is quite a large difference in the number of deletions, for example, and that's kind of dependent on the family structure. If the family have a slightly more distantly related family member, that can bring more unique variants to the kind of large melting pot for that whole family. So to summarize, we've put together a novel combination of tools to improve upon a previous pipeline for detecting copy number variants from family-based sequence data. And the selection of tools is very important for making a consensus call, because if we make a consensus call, we want some of the tools to support each other, but the flip side of that is we might lose the individual advantages of each of our tool. So what we're showing, and what the Canadol paper has shown, is that knowledge of family structure can definitely reclaim some of the advantages of each of our individual tools, but we need to be quite careful of CNVs in regions of maybe low mappability or high coverage, because some of the tools can be quite sensitive. So going forward, as Niamh mentioned, we'd like to apply our pipeline to the Tourette syndrome family, to all of our other families. We have quite a large database of data here, so it'll be very interesting to see across multiple families what we can tell. So we're quite excited for that. So I think that's us here. So we'd like to acknowledge all our collaborators from across the PGC, specifically Carol Matthews and Bill Byerly, whose data we've presented here. It's very fascinating work. We're all very excited about it. I'm sure we're very happy to take any questions, but also just to note that we'll be at the World Congress next week, and myself and Niamh have 3 posters. So if you'd like to talk to us about either the two we've presented here or our posters, then we'd be very happy to chat.
Thanks very much.
Carol, or not Carol, I should say tomorrow.
Have you lost Tammy?
I don't know.
Don't stop video.
I'm here, so I don't see any questions currently. If you guys would like to ask questions, please put them either in the chat box at the bottom of your screen or in the Q&A box in the bottom of your screen, either one.
Maybe put the presentation back up in case you have to go back to particular slides.
Okay.
There was a question about whether the slides will be posted and available. The video will be for sure posted on YouTube. Would you be willing to make the slides available?
Oh yeah, sure. No
Okay.
problem with it. Yeah.
So I can send a link to those slides. I'll put them in Google Drive and send a link out along with the link to the video once it's posted.
Great, thank you. Thanks, tomorrow.
I hope this means that everyone's happy.
You know, they're saving all the questions for when they see you in person.
Yeah, yeah.
The World Congress, yeah. So, if there are no more questions tomorrow, do you want to leave it?
Sure, one question just came in.
Okay.
Okay.
Were samples run on arrays as well to verify CNVs? And that's a question from Jim Crowley to all panelists.
So no is the short answer. That kind of was our original plan, just as in a screening method that we might run all of our samples in one particular family on an array first to, so we might calculate, for example, polygenic risk scores, if we could see if there were any very well-known CNVs present. We thought it would also be quite useful instead to really look at the full spectrum of CNVs. We thought it wasn't necessary for us to kind of know in advance if there would have been any CNVs.
Saying that we are talking about validating the CNVs that we've identified, and we're looking into using methods like nanopore technology, which allows you to look at larger lengths of DNA in one stretch, which should also help verify the CNVs.
Jim says, thank you.
You're welcome.
Okay, thank you.
Any other questions?
I feel it's the same.
So I don't see any more questions coming in. Aiden or anyone else, do you have any parting thoughts?
I guess if anyone does have any questions or thoughts, just email us, or if you have any suggestions, or if you have pedigrees that you're interested in talking about, just email me or the guys here. So I think our emails were distributed.
Earlier, yeah.
Or catch up with us at the World Congress next weekend.
Oop, another question came in.
Oh, okay.
This is from Elliot Reese. Do you plan to use the Nomad SV resource to focus on ultra-rare CNVs?
Yeah, so Nomad is an interesting one.
Yeah.
Up until 2 days ago, Nomad was only available in HD37 for CNVs and structural variants. So that's one of the examples where we've taken HD38 data and lifted it back to Nomad to look at CNV frequencies. Now that HD38 has come out, we're definitely going to go in and compare our CNVs to the full CNV data set.
So as part of our filtering process for removing common variants, Nomad is one of the databases that we use, as well as the DGV and the Decipher DDD dataset. But it's unfortunate that, you know, Nomad was only available on HD19, which means that when we lift over, we do lose some information, so we can't really conclude a lot. But,
so we'll be very interested to see if there's any changes to CMV frequencies when we consider the HD38 data versus the older build.
Elliot says cheers.
Thanks, Elliot.
It's a good question, though.
Good.
It's really important to talk about things like reference data sets, because that can really make such a difference on the list of variants that you're taking forward, particularly when you're interested in rare variants. And the idea of taking forward something that you think is rare that's actually common is quite a, yeah, it's not a nice thought. So we are as thorough as we can be in excluding variants that don't match our criteria.
Any other questions? I'm not seeing any additional questions, but as Aiden said, the email addresses of the panelists are available, and they'd be glad to field questions at a later date or anytime. What else?
Sometimes it's nice to go away and mull over a presentation before giving answers. So yeah,
Anyway,
we're
so
happy
look,
to leave it there.
yeah, we're happy to leave it there, and thanks very much. Thanks, Tamara, for your help with this, getting us up and running.
Yeah. Thanks for the opportunity.
And yeah, thanks for the opportunity to talk about the project. So we'll sign off. So thank you. Bye.